If you still didn't get that, I think you can figure it out. Instead of sitting there with your hands folded on your lap, actually give it a shot. You know that we're going to put letters in here, and you know that each parent contributes one allele. So if this is a baby, and this is a baby, and this is a baby, and this is a baby, they're going to get an allele from this parent, and they're going to get one from this parent. Okay? A Punnett Square is a great tool for planning out how the babies are going to turn out. Now, some of you can see underneath, I'm asking you to also predict what percentage of these 4 babies, we call them offspring, are going to have brown eyes. What percentage are going to have blue eyes? So if you've already done your Punnett Square, you can go ahead and figure out those percentages as well. How many have done all 3 Punnett Squares? I'm just curious. Okay. All right, I'm going to talk you through this, and I want you to check your answers. If you didn't get the percentages yet, you can still predict here in a little bit, but I want you to follow along. You know, if you watched the video completely, you saw where he showed about crossing purple flowers with white flowers, and he showed you how to use a Punnett Square. There's a more advanced one on the back of that paper. Like I said, we'll get to that tomorrow, but if you get the concept, you'll be on your way. Up at the top here, we've got the mom. Down here on the side, we've got dad, right? And both mom and dad are going to have babies. In a Punnett Square like this one, it's always 4. 1, 2, 3, 4 offspring that we can have. With me? Okay. And what this lets us do is predict the likelihood that those 4 babies will turn out a certain way. And what we're interested in are traits. And this particular Punnett Square is designed for traits about eyes. Now, this is a famous thing to use in genetics, but it's also wrong because it's not as simple as 2 alleles. Eye color is more complicated than this, but teachers have been using this since the beginning of time. So I got this from someplace else, and we're just going to go ahead and use it, but I want you to understand eye color is more complicated than just 2 alleles. But in this case, we have a dominant allele and a recessive allele. And when I say fill in the Punnett Square, it is as simple as whatever you see here, you write here, and you write here. Same thing here, big B, big B. Whatever you see up here, you copy down here. And whatever you see here, you put next to it. So if dad over here has little b here, it's going to be a little b here and a little b here. You see, we just filled it in, going across. Big B here, this is going to be big B, and this is going to be a big B. Does that make sense? That's all there is to filling in a Punnett Square. It's that simple. You just bring it on down. Now, there are some rules. You know, we're going to put mom up here today, dad over here. Sometimes people flip that, and I may ask you to put dad up on top. The other rule is if you've got a dominant and a recessive, you always write the dominant one first. You with me? Okay, so with that in mind, let's fill in this one. What's this going to be?
Big B, big B.
Big B, big B. And this one?
Big B, little B.
And this one?
Little B, big B.
Well, we always write the dominant one first, big B, little b, and this one?
Little B, little B.
Little b, little b. Okay? These, mom and dad get together, we're going to get a
Big little b.
Big B, little b,
Little b, little b.
little b, and
Big B, little b.
big B, sorry, I screwed that up. Little b and
Big B, little.
little b, little b. Okay. Now, once you do the Punnett Square, the answer's sitting there in front of you. All you have to do is interpret it. And oftentimes they'll ask you questions like, what are the chances of something happening? What are the odds? What is the ratio? And these are simple math things you should already know. But unfortunately, we get these words confused. And when we read the question, we don't look carefully for which one are they asking for. In the case of this question, I'm asking for the chance or probability. So I'm looking for a percentage. Okay? Sometimes they might ask for a fraction. And if I say it's 1 out of 4 chances, okay, what is that in percentage? 25%. If it's 3 out of 4 chances, what is that in percentage? 75%. And I hope you do understand that 3 out of 4 chances, a fraction, 3 divided by 4 is 0.75. And we move that decimal 2 places and we get 75%. They all mean the same thing. But when you answer stuff like that assignment from yesterday, you don't give me the answer that the question's asking for, you're going to get it wrong. Chance is a percentage. Odds is different. Let me explain. Let's take this first one. Tell me, how many offspring are there?
8. 4.
4. 1, 2, 3, 4. That's what the boxes represent, babies. Okay? Out of those 4 babies, how many have brown eyes? All of them. So what percentage have brown eyes? 100%. Okay? How many have blue eyes? 0%. So that makes it real easy. So let's move on to this one then. Out of the 4, how many have brown eyes here?
75%.
Well, that's got dominant allele, dominant allele. The dominant always wins, so that's 75%. How many have blue eyes? Well, that would have to be the remaining. 1 out of 4. 1 divided by 4 is 0.25 equals 25%. Okay? Now that's fine for probability or chance. What if I ask you, in this case, what are the odds that these 2 parents will have a blue-eyed baby?
Which ones?
Now, this one right here, second example. If I ask you what the odds of them having a blue-eyed baby, Chase? 25%. Okay, here we're not talking about percent. Percent was the last one. Here we're using the word odds. Now, if you've ever seen gambling, we always say the odds are blank in blank. So, what are we looking for? No, that would be a percentage. We're looking for odds. Chase? 1 in 4. Excellent. So in this case, we would say the odds are 1 in 4. Where do you get the 4? There's 4 chances. 4 times we roll the dice. And 1 out of 4 of those times, we come up blue. You see that? 1 in 4. Now, I point this out to you because if I'm asking for a ratio, it's a whole different ballgame. Okay? A ratio is comparing this group to this group. Okay? What I might say, for instance, what is the ratio of blue-eyed kids to brown-eyed kids? And what would you say there?
3 round to 1.
Yeah, I said, what's the ratio of blue-eyed kids to brown-eyed kids?
3 to 1.
Gabe?
3 to 1.
3 to 1. So for every 1 brown-eyed, there's going to be 3 other way around. Yeah, that's what I thought you meant. Okay, so for every 1 blue-eyed kid, there's going to be 3 brown-eyed. You see that? You got to write it the proper way. I said, what's the ratio of blue to brown? And so the blue number has to be first. For every 1 of these, we're going to have 3 of those. And the way we write that ratio, as you should already know, is like this, 1 to 3. Still with me? Okay, so when I ask for a ratio, this is what I'm looking for. They might ask for odds, but if they say percentage, specifically they say, what are the odds of, give it to me in a percentage, well, then you can give it in a percentage, okay? When I say the odds are 1 in 4, 1/4, well, that's 25%. So if I ask for a percent, you give me percent, okay? You might notice that when you do ratio, you add these two numbers together, it equals 4. The same number of chances you have. When you do odds, does that equal 4? No, okay? There's 4 possible. That's always going to be that second number. Okay, let's do the last one over here. So, in this one, what's the chance, the probability that we'll have a brown-eyed child?
50%.
50%. So half of those, 50%. So we would say, how many in 4? 2 and 4. Although that looks kind of weird, we probably reduce that to 1 and 2. It means the same thing. Okay? And what's the probability of having blue? 50%. Okay? So we would say for the ratio, if I said what's the ratio of brown to blue, you would say 2 to 2, right? For a total, or you could reduce that to 1 to 1. If you reduce it, though, it's no longer going to add up to 4. Just a little thing to remember. How many got all 3 of those right? The Punnett Square filled in right. Did anyone actually get the percentages done before I went through them? Did you do it correctly? Yes? Okay. Now, we're going to do some other practice ones on the dry erase board thing, but right now we're going to grade Mendelian genetics. So I need those 2 that were gone yesterday. Now is your time to take it outside. The rest of you, I need to trade those in our rows right now. Okay. Again, you can put these 2 papers, these 2 homework assignments, in your binder. There's something sitting underneath you there. Is that your paper that you didn't complete? Some calendar. I know some of you don't believe me because you didn't do anything in 6th grade, they passed you on. You didn't do anything in 7th grade, they passed you on. I'm not going to do anything in 8th grade, they're going to pass me on. But I will tell you guys, regardless of whether you go on to your freshman year, if you maintain the same work habit that some of you have, which is not doing any homework, or you do it and you don't bother to put it in your binder, you will find yourself failing next year. And by your sophomore year, you won't be a sophomore, you'll be a freshman because you'll have 0 credits and you will drop out. And the whole course of your life will change. It's time to put on your big boy and your big girl pants and start acting like a full-time professional student. We're not having a discussion about this. I don't have time to waste right now. So what I need you to do, if you didn't finish, guys, that's not my fault. That's your fault. I allowed you to stay after school. I was here this morning, so you could come in and work on it this morning. So don't give me the computer excuse, because I've heard them all. Yes, sir?
I did it, and I passed it back, and I didn't get one.
Okay. Who in that row is grading one that didn't have one? River? Would that be you? Okay. Guys, please, do we have to waste my time with this? Let's see. You got it? Did you have one? Yes, that's it, right there. And you have Phillips. Where's yours? Oh, wow. Interesting how that works. It just stays on your desk, and then suddenly it shows up on my stack with all the right answers. Yes, sir?
I need a pen.
I don't supply pens or pencils. It was interesting speaking with Miss Rowe and Ms. Valdez. I guess I'm not the strict one when it comes to pens or pencils. She requires that you have 3 sharpened ones. You don't get up and sharpen during class at all. All I'm asking is you to have 1 working writing implement that's black blue. And some of you can't manage to do that. Rated by your name on the bottom, please. All right. This is going to be out of 27 points. Here's what's going to happen. I'm going to bounce around. We're going to go from grading to putting stuff in our notes to showing examples. So it's not just grading, but you need to pay attention. Each response is worth a separate point. Who is this dude we've got pictured up there? Gregor Mendel. Okay? I don't care about the spelling, although, really, guys, it's right up there in the word Mendelian. Mendel. And he did experiments with the pea plant. I'll bet some of you wrote P-E-E.
I did.
I don't want to eat P-E-Es. I would mind P-E-A, but if you wrote P-E-E, mark it wrong. You know the difference between P and P. Okay? On the next one. When Mendel crossed pure purple flowers with pure white flowers, he got what? Purple. So this monk that did all these breeding experiments with the pea plant, you know, he set the foundation for modern genetics. And when he took a purebred white flower and purple flower and bred them together, and all the babies were purple, he was a little surprised. You would think that some would be purple and some would be white, like maybe half, right? That makes sense. Or you think that purple and white would give you pink, but that's not what happened. And that little discovery is what led him to start to think about things like, how are you going to find yourself out here? Things like how one allele might be more important than another allele. How one allele might cover up what another one is showing. What is that called? What's it called? Dominance. The one that's being covered up is called recessive. He's the one that coined all these phrases. This idea of the two alleles. So we started with purple and white gives us all purple. I wonder why. He figured out that the purple covers up what the white wants to show. Okay? Now, one of the tools we use for planning how the offspring are going to turn out is the Punnett Square that we just did. Another tool we use is called a pedigree. And I want you to write these two definitions down in your notes right now. Punnett Square and pedigree. A Punnett Square, and you should have already written the words down, all you have to do is do the definition, is a tool that helps to show all possible combinations of alleles. A Punnett Square is a tool that helps show all possible combinations of alleles. And it might not be a bad idea to just draw one over there. A tool that helps show all possible combinations of alleles. A pedigree, well, that's where you get one of these things, like what I drew on the board. A pedigree is a diagram that shows the appearance of a particular trait as it is passed down through generations.
Oh, is that like how the monkey got to the
Yep. These pedigrees all the time, just like in evolution, they'll show this came from this. It shows heredity. You feel this is too hard for you? Yeah.
Oh.
You're sitting there with paper and not writing. A diagram that shows the appearance of a particular trait as it is passed down through generations. Like this. Like that one. So you might notice I underlined a key term. Punnett squares, it's alleles. Pedigrees, it's traits. Did you get this down, Ethan? Doesn't look like it. So don't care about notes? Don't think you need them? Don't have anything to write with.
Oh, I have something to write with.
So when we're doing the Punnett Square, we're looking at the big B, little b. We're looking at the parts that go into it. But we're more interested often in the genotype. What do we have? Big B, little b, big B, big B. When we're looking at the pedigrees, we're more interested in the trait. Okay? This particular person has the gene for Huntington's, and he's going to pass it on to one of his kids. Which one is most likely to get it? What are the odds? This type of thing. And we look at traits. Yes?
If Huntington's is what makes you shake, and what is like Parkinson's? Doesn't that make you shake?
I don't have time to get into that.
What?
I don't know about the genetic components of Parkinson's. He asked about Parkinson's. Well, there are a lot of nerve disorders that break down how the nerve works, how that sheath goes around the outside, and some of them are definitely inherited, genetically linked. Okay? All right, so this is what Mendel did, and I could show this in a pedigree. We have purebred purple, purebred white. Put them together, they have babies, and look, not a white one in the bunch. And the reason for that has to do with the alleles. But this is the way he could keep track of things. You see this purebred purple, big P, bred with a white, which is little p, little p. And we all know that if dad has to give one allele to each of these, it's going to be purple, right? Because that's already dominant. It's going to win every single time. But even though the mom's allele, if you will, the white flower's allele, doesn't show up in the phenotype, it's still there in the genes, the genotype. And what we see down here at the bottom is, okay, now let's just say you bred those two flowers together. Well, we could get big P, we could get a flower like that. Or we could get little p, and we could end up with a white flower. And what happens is, when you have a purebred dominant and a purebred recessive, and they breed, that one recessive trait doesn't show up in that first generation of babies, Philip. But it will show up in the next one. And there's a term for each one of these generations. This first generation of the two parents is called the P generation. What do we call the second generation? The babies from that first crossbreed.
F1.
F1. What do you think we call this one?
F2.
F2. So there's terms for each one of those. And when we breed a purebred dominant with a purebred recessive, we don't see that recessive trait showing up until the F2. It skips a generation. And I showed you why that is with the alleles. But on a pedigree, we're just looking more at the traits. Okay? So now that you know that, let's go back to this and get the answers, see if they were right. The P generation was the parents. I'm sorry, I'm really pressed for time. Question?
What if they go purple flower?
That's fine, as long as the word purple is there. Okay? The P generation, or P1 is the first one, the cross of the two parents. And then we get the F1 generation. And of course, after that would be F2. In that F2 generation, okay, so we parents, babies, babies of the babies, the grandbabies of that flower. Okay? You saw that 3 of them were purple and 1 was white. That's what happens in that situation every time, okay, when it's a dominant recessive trait like that. 3 to 1 is the ratio. For every 3, there's going to be 1 with recessive. So that's 2 separate points. Please score that as 2 separate answers. 3 to 1. All right, moving on. Mendel's 2 laws are the laws of segregation and the law of independent assortment.
3 points.
3 points. 1, 2, 3. And yes, they have to be in that order. Okay? Now, what are these laws? Well, segregation means that there is a 50% chance of getting either allele. Okay? My dad has 2 alleles, my mom has 2 alleles, each one passes on 1. I have a 50% chance of getting this one from my dad or this one. 50% chance, because there's 2. That's what they mean by segregation. Those 2 alleles that determine what traits you're going to have work separately. Okay? Segregation means to be separated. The law of independent assortment, this next blank, is basically saying that, at least in the case of what Mendel looked at the gene for seed shape and the gene for flower color have nothing to do with one another. They're unrelated. Just like your IQ and your height are not related. They're two separate genes. We say they sort independently. Okay? So they're independent of one another. That's what he learned from that. Yes, Kandari.
So like the red hair and freckles, what do you call it?
Perfect. Thank you. I was just going to say that. So that would be an example of where they're not independent, right? And that's why that question says, except in the case of linked genes. So red hair and freckles would be linked genes. And the truth is, guys, there's lots of things that are like that. There are some that work with one gene. There's some that are linked by sex. There's some that are linked to others. Yes?
Does that mean like there's a 100% chance? Like if you get freckles, you have to have red hair?
No.
Or if you have red hair, you have to have freckles?
Not that one. I don't know. I don't know. Okay.
So it might be like...
I don't know. I don't know. So the next one is a good thinking question. So let me just throw this at you. If I flip a coin, what are the odds it's going to come up heads? What are the odds it's going to come up tails? 50%. What if I flip it 10 times, and 10 times in a row it comes up heads? Does that make it more likely that the 11th time it's going to come up heads? No. It is just dumb luck. And the thing is when we're looking at Punnett squares like these, and we see, oh, this person has a 75% chance of having brown-eyed kids. And then you go and you have this genotype, Philip. You have this, your wife has this genotype, and you have babies, and lo and behold, only one of them has brown eyes. Okay? Does that mean this was wrong? No, you just need to have more babies. What if you only have 2 babies, and they both have brown eyes? See what I'm saying? Okay, you took care of these. But if you keep having babies, the odds are that one more will have brown eyes and one will eventually have blue eyes. But you might have to have 16 babies before that percentage holds true. Charlie?
So I have 9 siblings.
Genetic siblings? From the same parents?
Except for 2 of them.
Oh, okay.
And all the dads have blue eyes, and
Okay.
my mom has brown eyes.
And all the kids have brown eyes?
Except for one of them. One has blue and one has green.
Okay. So again, remember I said that when it comes to eye color, there's more than just 2 alleles. It's more complicated. In general, brown eyes dominate over blue eyes. This is not accurate truly for eyes. I could change this from eyes to hair color. Blonde hair is recessive to brown, okay? But there are more factors playing into that with other alleles, but you will see a predominance, way more brown eyes than blue eyes. And the blue, and I think the green or hazel are kind of lumped together. Yes?
I heard that like, how that works, it's like flipping a coin, but you don't get like 50% like all the time. They said that you have to do like thousands of times before you can match up.
Right. Okay, so even though I got, that's good, thank you, Jerry. Even though I got that heads 10 times or 40 times out of 50, doesn't mean the 51st flip is more likely to be heads. Basically, coins don't think, right? You flip a coin, if it gives you heads, it doesn't influence what the next flip is going to be. Okay? It's the same thing with this. The law doesn't change just because you got 2 brown in a row doesn't mean the next one has to be brown. Yes?
So you just kind of explained the eye color thing, but my mom and my dad, they both have brown eyes, and then they also both have a widow's peak, and I don't have.
Unrelated.
either.
They both have a widow's peak,
Yeah,
like the pronounced one?
yeah.
Maybe yours is still forming. I don't know. Okay, so the answer to that one was 50. Which of the following are homogeneous and which are heterogeneous? I'm sure some people got these wrong. Kandari, spit it out, please. Because, whatever you turn on.
My name. It was.
Yeah, I saw you swallow it too. He wasn't here for the whole part about how the epiglottis works. All right. Little b, little b. Big B, little b, big P, little p. These are 3 separate answers. I don't care which of these is first here, but if they use different letters, they didn't follow directions. Notice, I said this part was not going to be identical. Please stop that, Aiden. Okay? These are heterogeneous because the 2 alleles are different. These are homogeneous because they're the same. Purple, purple, white. Purple, purple, white. No other options are correct. Not pink, not purple/white. When you have 2 for purple, you're going to be purple. When you have purple and white, you're going to be purple because this one's dominant. And when you have little p, that's the only time you're going to show that white. That's why it only shows up 1 out of 3. Okay. When we are crossing a big P, little p with another one of the same, the offspring will be purple 3 in 4 times. 3 in 4. That's the only number that's correct there. There's 4 outcomes, 4 babies. 3 of them will be purple. That one that's not is the one that's white. Okay, then the last part. Should have been straight out of the video. And we're talking about Guthrie having Huntington's disease, this terrible disease that causes degeneration of nerves and eventually leads to death. He knew he had the gene, he died from it, and his son had a 50% chance of having that gene and becoming sick and dying early, a painful death from Huntington's. So the question is, do I go, because they developed a test, do I go and get tested and find out that I'm going to die painfully young? Or do I just cross my fingers that it's not going to be passed on to me and not know? And if he does get tested, then do we tell his insurance company? Because they're not going to pay for his insurance knowing that he has this terrible disease. You see, that's one of the things we face with this. A pedigree, like I showed you over there or on the board, we always represent the guys with squares, and the things that are connected by that horizontal line are the offspring, the baby. Okay?
It's like a family tree, just shaped.
Well, that's exactly, it is like a family tree. That's exactly what it is, except we use specific symbols, and we're trying to show traits. Not just who your parents or your siblings are, but traits. Philip? If this isn't going to work, I'm going to have to move him again, and I don't want to do that. Okay? All right. Whether or not to be tested, that's what we call an ethics issue. We're going to spend a whole day on genetic ethics because there's a bunch of them. Things you have to consider. Is this the right thing to do? Score that out of 27, please, and then pass those up. 27. All right. Here's what we're going to do, guys. I don't have time to waste, so let me get your attention up here. So far, we define Punnett Square and pedigree. If you were outside, you'll want to watch the podcast or get those from a classmate. Punnett Square and pedigree. Now you're going to look for these 2 definitions. Codominance and incomplete dominance. River, back up 1 seat, please. Codominance and incomplete dominance. You'll see them in this little video. He'll talk about it, and then he'll pause and give you a chance to use that information in a Punnett Square. That's why I gave you the marker, the eraser, and the whiteboard thing. Okay? So, 2 things. Fill in the Punnett Square when it comes up, and write down the definition.
In our notebook?
In your notes, yep, the ones you set up.
Okay, so the topic of this video is going to be the genetic inheritance pattern known as incomplete and codominance. We're going to do 2 patterns at once. And you'll see the two patterns have some features in common with one another, but also some key differences. So let's go ahead and get started. So let's first look at the inheritance pattern called codominance. And the definition of codominance is when a single gene has more than one dominant allele. And if you've seen my other videos before, we've never seen this pattern before. And this was completely unknown to Gregor Mendel as well. If you recall from Mendel's experiments, he was looking at, for instance, flower color. The peas either had purple flowers or white flowers. Purple was dominant, white was recessive. But in codominance, it's possible to have more than one dominant allele. And for instance, an example is in the cattle coat, or the fur of cattle. For instance, in the picture, we have an individual with red fur and an individual with white fur. Now, when you look at the red cow and the white cow, if they were to breed and reproduce with one another, would all the babies come out pink? That'd be adorable if that was true. But no, that's a myth. That's not what happens. So what does happen in codominance? Watch this. What we see is a pattern, a reality where both colors are equally expressed. You see like patches of red and you see patches of white, and we call this color pattern here roan. So red's not dominant, white's not, I should say red is not dominant to white, and white is not dominant to red. They are equally dominant. You see both at the same time. A good example of codominance in humans is in blood types. You might have heard of different types of blood, like blood type A, blood type B. Well, when we look at blood types, here's a red blood cell, and blood type A has these proteins on it. Now, the proteins are not actually shaped like English letter A's. I'm being a little simplistic here, but blood type A has some proteins on the surface. Blood type B has some different kinds of proteins on the surface. So what happens if mom was blood type A, and what happens if dad was blood type B? If mom and dad were to produce a child, for instance, the two blood types would be codominant. The child or the offspring that was born would have characteristics of both blood type A and blood type B. So we're going to go into this in a little more detail. So let's talk about blood types now. When it comes to blood types, there are 3 alleles, and this is something that we haven't seen before. When Mendel was studying his pea plants, for instance, he was studying the gene for flower color. There were only 2 alleles. Flower colors were either purple or white. When Mendel was examining the gene for seed color, the seeds were either green or yellow. There was never a third option. But in blood types, there are. There's an A allele, which happens to be dominant. There's a B allele, which also happens to be dominant. And there's an O allele, which is recessive. Now, when we look at the picture on the right-hand side, you see the box that said mom will pass one of these 3 alleles onto her offspring. Maybe mom has type A, maybe she has type B, maybe she has type O, but either way, mom is going to pass on one of these 3 alleles to her child. On the top of the picture, dad is going to pass on one of these 3 alleles to the child. So when you look at what are the possible genotypes that a child can have when mom and dad have a baby with regards to blood type, these are the 9 options. And when we start going through the blood types, 2 capital A's, that's type A blood. 1A and 1B, that's codominant. That's codominant type AB blood. The next one to the right, dominant A and a recessive O, that's blood type A. A is dominant, O is recessive. When we look at this, the middle row, if you inherit 1 dominant A and 1 dominant B, then that's blood type AB. The next box to the right, if you inherit 2 B alleles, your blood type will be type B. The next one to the right, if you inherit 1 B from mom and 1 O from dad, blood type, you'll have blood type B. Again, B is dominant, O is recessive. And finally, the bottom layer, or the bottom row of this table here, if you inherit a dominant A from one parent and a recessive O from the other, you'll have blood type A. Blood type A is dominant, blood type O is recessive. Same with blood type B. B is dominant, O is recessive. So the only way you can have type O blood is if you inherit not 1 O allele, but 2 O alleles. So what we learned from looking at blood types is that blood type A and B are dominant to blood type O. Blood type O is recessive. So therefore, the only way to have blood type AB is if you inherit a dominant A and a dominant B. They are codominant. They are equally dominant. If you inherit 1A and 1B, A does not cover up B, and B does not cover up A. They are equally dominant to one another.
All right, so let's look at that example. He's going to give you one here. Hopefully you got down your definition of codominance, when a single gene has more than one dominant allele. Okay. By the way, one of you passed up the 2 homework pages. I asked you to put that in your binder. If it's you, raise your hand and I'll give it back to you. But you should have those 2. Okay, so codominant, think of like co-captains. When you are co-captains, you're both in charge. It's not like one of you says, oh, I'm co-captain, so you shut up. No, you both get to talk, you both get to contribute. And in the case of the blood types you saw, you can get an A allele, you can get a B allele. When they come together, you get something called AB. They both have their voice heard, so to speak. They both are seen in the result. And that's why we call that kind of blood AB. Unlike when we have, big W and little w, the big W masks, it hides the little w. And if this were widow's peak, we're going to have it. This is going to hide that one, okay? This is not regular dominance, it's codominance. So both of them are seen. Now we still have a recessive. We still have the little o. So hopefully you'll be able to see up here the blood types are A, AB, O. But there's different versions of A. 2 different versions of A. Do you see that? And there's 2 different versions of B. But whenever we say your blood is codominant, you automatically know what type it is. It's AB. Do you see? Because it's the only example where we have 2 different dominant alleles. These aren't going to fight with one another, so we don't call it codominant. 2 different alleles. So, here's the problem that he throws out at you. Mike is heterozygous for type A blood, and Lisa is heterozygous for type B blood. And then it wants you to fill in the Punnett Square and answer some questions. So I want you to try this out. I'm going to help you. Since this is your first one, I'm going to show you the thinking process, how you set it up. Mike, the dad, we're going to put dad over here. And it says he is heterozygous. And we already know Mike is type A. The question is, which one is it? He could be this type A, or what's the other option? No, AB would be AB. Big A,
Little B.
little O. There is no little B. Did you see a little B on the last screen? No, there is no little B. If you have type B blood, it's a dominant allele. So these are the 2 options. So before we can fill in dad over here on this side, we need to know which one is right. And the secret is right in here. If he's heterozygous, which one must it be?
It's A.
Is it AA or AO? AO. How did you know that? Heterozygous is mixed. It's different. They're not the same. These 2 are the same. That would be called homozygous. So we're going to put the A here and the O here. Remember, the O is recessive. Now, Lisa, the mom, is up here. And Lisa is, it says, heterozygous for type B blood. But again, type B could be big B, or big B, little o. It can't be AB, because type AB is not type B. Those are different. AB is type AB. Which one is right for Lisa? It has to be this one, because it says hetero, and they have to be different. So we put her B here and the little o there. go ahead and fill in the Punnett Square and see if you can come up with these percentages for me. The first question, and all these are looking for percentages. When I say probability, the first question says, what's the probability of having a child with type A blood? Remember, type A could be like this, or it could be like this. The second question is, what's the probability of having a child with codominant blood? Well, you already know what type that's going to be. What's the only type that's codominant?
AB.
AB. And then the third question, these aren't the answers, these are just hints. The third question is, what's the probability of your child being heterozygous? So we're looking for when the two alleles are different. Yes? You got all of them answered?
Yeah.
Well, I don't see them down there. I want 3 percentages. Percentage A, percentage B, percentage C. You gonna write them down? Percentage A, percentage B, percentage C. Who thinks they've got them already?
I got them.
Show.
Is number 1 25%?
Number 2,
0%.
number 3.
50%.
Okay, what else we got?
25.
All right, 25, 0, and 50. What'd you get? 50, 25, 25. We got 50. Okay, so let's fill this in and let's see.
Can I do mine?
Wait.
Yeah, what's yours?
Question 1 is 25. Question 2 is 25. Question 3 is 75.
Okay, let's find out if you're right. So we just fill these in straight across. A goes here, A goes here, O goes here, O goes here. Remember, we put the recessive 1 second. Then B goes here, B goes here, O goes here, and O goes here. So this first one, I'm looking for type A. So it's either AA or AO. That's not it. That's one. And that's the only one. So if it's 1 out of 4, that would be 25%. How many had 25% for that? Good. Okay, the next one, codominant blood. Oh, it's the only one that has 2 capital letters together that are fighting, that are different. Okay? And that would be this one here. AB. AB. So 1 out of 4 again is 25%. So far, you're 100%. What was your last prediction?
75.
75.
Yep.
Heterogeneous. Okay, so here we're looking for a mix. Mix, mix, mix. That's not a mix. Those are the same. So is he right?
Yeah.
75%.
Well,
Okay? Erase your board. Sorry. I want to answer, but we're going to run out of time here. Next one. I want you to understand this one right here. This says Jason is homozygous for type A blood. Okay? So Jason is the dad. And we know he's type A. And the mom, Maria, is heterozygous for type B. So this is the mom, Maria. And she is type B. But now we need to know what their alleles are. For Jason, if he's homozygous and he's A, has to be AA. Homo means same. These two have to be the same. Maria, heterozygous for B. So, could it be BB? No, it has to be BO again. Remember, heterozygous means mixed.
Can it be BA?
No, because type BA, it's actually said type AB, is a whole other blood type. AB is a blood type. Okay? She's not type AB. She's type B. Yes?
Would AB still be heterozygous since they're mixed?
Yes.
Would AB be what?
Heterozygous.
Heterozygous? Yes. Yes, it would be heterozygous, even if it is codominant. Okay, so now, when we got our alleles in place, we find AB here. Is that what everybody got, I assume? AO, yes? AB. Sorry, I don't have time for you to hold them up right now, but AO. So if I ask you what is, well, it says the son here, Rick, is codominant. And if we were doing a pedigree, we would do something like this. Here's Jason, married to, oops, a circle, Maria, and they have a baby. This is a pedigree, and he's Rick. And Jason has type A, and Maria has type B, and it says Rick has codominant blood. So you already know what his blood's going to be. Are we getting inappropriate on the boards there? What's it going to be?
AO.
Would that be codominant? You need two big letters for AB. So we already know he's going to be AB, right? Well, what's the chances of him with those two parents getting AB? 100%?
No, 50.
50%, right? 50% was AB. The other 50%, what type is that going to be?
Dominant.
If the A is dominant, what type is it? It's type A. The A is dominant, it's type A. Okay, you got one more definition to get in there. And this is on incomplete dominance. Okay, looks like this. So previously, we saw red and white both made their voices heard. That cow was not pink. The cow had part red and part white. Now, we have something quite different. It's almost like a compromise. The two features are blended. They're blended. So instead of the red totally dominating the white and covering it up and masking it, okay? Instead of them sharing equal roles, they blend together. It hasn't completely dominated it. The white hasn't totally covered up the red, and the red hasn't totally covered up the white. That's how it got its name, incomplete dominance.